Imagine a world where genetic diseases like cystic fibrosis or Huntington's could be erased, not just treated, by literally rewriting the faulty code in our DNA. This isn't science fiction anymore. It's the promise of CRISPR gene editing. But with the power to edit the very blueprint of life, we face incredible opportunities and profound ethical dilemmas. Could we truly eliminate these conditions forever? And if we could, should we? Everything is a system. Understanding complex biological processes like CRISPR gene editing and its potential to cure genetic diseases requires a systems thinking approach, especially for grade 11 students exploring advanced biology. We'll explore how different parts of this powerful technology interact to determine its effectiveness and safety. CRISPR-Cas9 is like a molecular GPS and a pair of scissors, explains Dr. Anya Sharma, a lead geneticist at the Broad Institute. A guide RNA component directs the Cas9 enzyme, our molecular scissors, to a precise location in the DNA. It finds a specific sequence, then cuts both strands of the DNA. The cell then tries to repair this break, and we can trick it into inserting a corrected sequence. The challenge lies in ensuring that our guide RNA specificity is perfect, that Cas9 cutting efficiency is high, and that the cell chooses the right repair pathway selection. To truly understand CRISPR, we need to look at its key components. Our external inputs are guide RNA specificity, which determines how accurately the system targets DNA, and CRISPR efficiency, how well the CRISPR components get into the cells. The internal outputs include Cas9 cutting efficiency, how often the DNA is cut, off-target edit rate, unintended cuts, repair pathway selection, how the cell fixes the cut, the Cas9 plus therapeutic benefit, the actual performance, the actual improvement in health, and unintended mutations, any new, unwanted changes. Let's see how these components interact. There's a negative relationship between guide RNA specificity and off-target edit rate. Higher specificity means fewer unintended cuts. Conversely, delivery efficiency has a positive relationship with Cas9 cutting efficiency. More components delivered means more successful cuts. A higher off-target edit rate has a positive relationship with unintended mutations, meaning more unintended cuts directly increase the risk of harmful new mutations. Finally, a strong positive relationship exists between Cas9 cutting efficiency and therapeutic benefit, as more successful corrections lead to better patient outcomes. These relationships highlight the delicate balance needed for safe and effective gene therapy. Build your CRISPR gene editing model. What if we could improve guide RNA specificity to nearly 100%? How would that impact off-target edits and unintended mutations? Or what if we developed a delivery method with perfect efficiency? How much would that boost therapeutic benefit? The field of genetic engineering is creating exciting new career paths. Gene therapy scientists, earning between $95,000 and $180,000 a year, design and test these cutting-edge CRISPR-based treatments. Meanwhile, bioethicists, who can earn $70,000 to $140,000 annually, advise on the profound ethical considerations surrounding these powerful technologies. Everything is a system. Model it, build it, simulate it, understand it. Visit ModelIt K12.com.